Bismillahirrahmanirrahim. Assalamu alaikum. This video is on fatty acid synthesis. Actually, I'll make two parts of the video so that you can properly understand, but you have to go through both parts. Okay, let's start. Fatty acid synthesis is a cytosolic process. What are the main sites in the body in which fatty acid synthesis takes place? Adipose tissue, liver, and in lactating mammary gland. What are requirements for fatty acid synthesis? It's an energy consuming process, so it requires ATP, it requires NADPH as a reducing agent because there are two reduction reactions in fatty acid synthesis, and the main substrate is acetyl-CoA. Acetyl-CoA is a 2-carbon compound which results from oxidative decarboxylation of pyruvate. What is pyruvate? It's end product of aerobic glycolysis. It contains 3 carbon. Then pyruvate undergoes oxidative decarboxylation in the presence of vitamin B1 in mitochondrial matrix to form 2-carbon acetyl-CoA. Then the question arises, what are the fates of acetyl-CoA? The fates of acetyl-CoA we have seen in previous videos. You can check in carbohydrate metabolism. Okay. The acetyl-CoA can enter ketogenesis, it can enter cholesterol synthesis in fatty acid synthesis, or it can participate in Krebs cycle. It also facilitates gluconeogenesis. So there are different fates of acetyl-CoA depending upon body nutritional and energy states. What is the rate limiting step of the fatty acid synthesis? It is a conversion of acetyl-CoA into 3-carbon malonyl-CoA. Here you can see. This is a rate limiting step, acetyl-CoA carboxylation to form malonyl-CoA. In the presence of enzyme is simply acetyl-CoA carboxylase, and coenzyme will be vitamin B7 biotin. 1 carbon will be added in the from carbon dioxide to form 3-carbon malonyl-CoA. So this is the enzyme, is called regulatory enzyme of fatty acid synthesis. So there must be some substances or conditions that act as activators, and there will be some inhibitors as well. I would like to tell you that acetyl-CoA carboxylase in its phosphorylated form is inactive. It means its dephosphorylated form is its active form. Dephosphorylated form is its active form. It means all that conditions and all that hormones which will result in dephosphorylation of this enzyme will result in activation of this enzyme. Okay, and that hormone will include insulin, because insulin inhibit adenylyl cyclase enzyme. If there will be inhibition of adenylyl cyclase enzyme, no cyclic AMP, no protein kinase, and no phosphorylation. So dephosphorylated form of acetyl-CoA carboxylase will be its active form. And in a similar way, glucagon, glucocorticoids, and cortisol, glucocorticoids, glucagon, and all such hormones that raise blood glucose level will cause phosphorylation of acetyl-CoA carboxylase and thus inhibition of acetyl-CoA carboxylase. So this is regulation, brief and simple regulation of acetyl-CoA carboxylase. Okay, what is initial requirement to start fatty acid synthesis? We know that acetyl-CoA results from oxidative decarboxylation of pyruvate in mitochondrial matrix, but fatty acid synthesis is a cytosolic process, so this acetyl-CoA must come out of mitochondrial matrix into cytosol. But the problem is same that was in beta oxidation of fatty acids, which we have seen in previous video, that carnitine shuttle was required for fatty acid beta oxidation because coenzyme A part was unable to cross in a mitochondrial membrane. The same case here, coenzyme A part is unable to cross in a mitochondrial membrane. So inside mitochondrial matrix, what will happen? This 2-carbon acetyl-CoA will combine with 4-carbon oxaloacetate to form 6-carbon citrate. Enzyme is simply citrate synthesis. When citrate level in mitochondrial matrix elevates, so it simply diffuses out of mitochondrial matrix across mitochondrial membranes into cytosol. What will happen in cytosol? This citrate undergoes simply lysis in the presence of ATP dependent citrate lyase enzyme. Citrate lyase. So citrate will undergo lysis to form again 4-carbon oxaloacetate and 2-carbon acetyl-CoA. Then this oxaloacetate can enter either Krebs cycle, either gluconeogenesis, but the more likelihood of this oxaloacetate is to enter Krebs cycle. The reason for this is that we know that fatty acid synthesis is an energy consuming process. So there must be a sideway process is required that is supposed to produce continuous ATP. So Krebs cycle will produce ATP, and that ATPs will be utilized for fatty acid synthesis. And what will happen to this acetyl CoA? It will undergo carboxylation to form malonyl CoA. That is the rate limiting step of fatty acid synthesis. Now, coming to the main fatty acid synthesis steps. So there are 7 steps of fatty acid synthesis, and the mnemonic is ATMCRDR. A stands for acetylation, T for transpiration, M for malonylation, C for condensation, R for reduction, dehydration, reduction. And I will tell you the complete detail of these 7 parts in the next part of the video. Okay, Allah Hafiz.